review, you guys remember how to number your carbons on your carbohydrates?
Yeah.
Remember that's carbon 1, this is carbon 4, 2, 3, 5, and then 6 would be, that's not right, but I'm just saying. Alright, so 6 would be there. Remember the way that you can tell if it's alpha or beta was high. How? So there are alphas.
If it's going up or down.
So which one is up and which one is down?
Alpha is down.
So alpha, remember, on carbon 1, the hydroxyl group will be facing down. So it would be the opposite for beta. It will be facing up, just as a review, just so you guys can review. All right? Remember that the bonds that are formed here, these bonds are hydrogen bonds. Yes, I'm sorry, I'm smoking crack. All right? And then remember that when you have your monosaccharides that are, if it was a reaction, that this would be, as question 1 says, name the type of bond indicated by T, and T is that bond between this and this, so what type of bond was that? Glycosidic bond, right? We think of glyco, remember glyco is sugar, so think of glycosidic bond as connecting 2 sugars together. Glyco is sugar, so think of it as glycosidic bond as connecting 2 sugars together. Okay? Because that's what glyco means. Alright? Dottie 2 says state the given name of the type of reaction in which water is involved. Hydrolysis. Because remember, they kind of give you. Yeah. Remember hydrolysis? Hydro is water, and it says which water is involved. Yeah. Yeah. So it can either be a condensation or a hydrolysis reaction. How do we know that this is a hydrolysis reaction?
Because we're adding water instead of getting it.
Yes, because remember, hydrolysis, water is added. Condensation, water is released. Just think about it. Think about it as like when you're looking at your glass of, well, not you guys. You should be having a glass of adult beverages. When you're looking at your glass of soda, the water that forms on the outside, remember that's condensation because water is being lost. Okay? So state the 2 roles of water within plant cells other than taking part in the breakdown of reactions. Somebody tell me what was one of the things you got.
Solvent.
It is a solvent. Yep. What else?
Helps the plant from wilting.
Yep. It helps to maintain makes the plant turgid or helps to maintain its shape. What else? A medium for what? Be more specific.
The transport chemicals.
Okay, so it is for transport medium. You just can't say medium. That's why you have to be very specific. Okay.
Chemicals.
So if you said that it was a medium, like water is used as a medium, you have to be very specific and say that it's a transport medium. Okay. What else is water used for in plants? How does plants take up water? From the roots, right? And it does that because of that water potential gradient, right? Yeah. So remember that water acts to help maintain the water potential gradient. Okay? So it's also used in when the stomata is opening. To open the stomata, you use water. So you could just pretty much say basically anything. You can say that it's a transport medium. You can say it's a solvent. You can say that it's used in chemical reactions because, or you could have restated again that it's used in hydrolysis and condensation reactions. So you can say a lot of different things when it comes to this one. It was only worth 2 marks. Yep. So you should have been able to get it. And it says enzymes are globular proteins. State what is meant by the term globular. Misa.
protein where the molecules curl up into a ball shape?
Yep, so globular means that it's ball shape. What level of protein is the globular shape found in?
Tertiary.
Tertiary, right? It's water soluble.
What's
Right.
malleable? It can malleable.
Yeah, like it can move to fit
Yeah.
the shape. Yeah. So what it means by globular, it fits into a ball. You could say that it's a tertiary structure. You could say what Wadley just told us, how it's able to move and to adapt or be malleable, and that it's water soluble. You can't say that it's more than one protein, because secondary is more than one protein, and so is quaternary, so that wouldn't be one that you could use, okay? It has to be something that is characteristic of just tertiary proteins only, okay? Or if they ask you about like secondary structure proteins, you have to state that what's just specific to those only, okay? Alright, and then C says the reaction shown in figure 4.1 is catalyzed by enzyme sucrase, as shown as the reaction. Name the part of the enzyme labeled U. Everybody better get that right.
Active site.
Active site. Okay? It's the active site. So, and then it says, in reference to the enzyme, it says states the mode of action. Whenever they ask you to state the mode of action of something specific, they're talking in general. Like, what's the function? How do enzymes actually function? So give me some ways that enzymes function.
Substrate connects to active
Substrate specific to an active site.
Catalyzes reactions.
Be more specific. It catalyzes reactions without what?
changing the
Shape. Oh.
Without being used up in the reaction, right?
Yeah.
Right?
It
So
enhances
enzymes,
the
yeah.
changes, so.
Wadley.
It forms an enzyme substrate compound.
It's from the enzyme substrate complex. What else?
Finalizing the two product
Or
molecules.
more.
You could have put any of the following. That it's specific, like the substrate is specific to the active site, or the enzyme is specific. That it only binds to it only binds with the active site. So it has a complementary or matching shape. So they give you a way that you can say in 3 different ways that it's just specific. Okay? You could have said something like the lock and key. Remember that? As Wadley says, the enzyme substrate complexes form. It lowers the activation energy. That's what enzymes do. It's not used up in the reaction, and that it catalyzes more. It speeds up the reaction to make it same thing, that it lowers the activation energy, but it also lowers the amount of time that a reaction needs to take place. Because remember, that's the whole point of an enzyme. It's to lower the amount of energy needed and to speed up the reaction. Anybody still confused about enzymes? So, if I gave you a graph, and I said to you, and I gave you arbitrary units, and I say to you, where's the reaction at its optimum efficiency? Yep. So here, right? This is when the reaction is at its top efficiency. This one we have the enzyme to substrate complex is equal, right? So here do we have more enzyme than substrate or less?
Less.
More enzymes.
More. Remember, as you start, you have more enzymes free. Yeah, you have more enzymes looking for something. And then here, either the reaction, all the enzymes the reaction has done, or it depends on the type of inhibitor you have, will depend on what's going on here too. But you need to be able to like extrapolate data points from off of it and know. By the way, independent or dependent?
Independent.
Independent.
Remember that?
Try that.
Oh, yeah.
Remember dry mix?
Dependent what?
Remember that?
Independent,
So the x-axis is independent, and it's the variable that you manipulate. The y-axis is dependent, and it responds. Just in case they ask you that. That's something that you should know from. Yeah. So just remember dry mix. Remember that. Yeah. Okay. All right. Any other questions about enzymes? Everybody understand the difference between competitive and non-competitive when we went over?
Yep.
Can competitive be reversed?
What do you?
Yes.
Yes. Why can it be reversed?
It doesn't each other.
Huh?
It doesn't denature the enzyme.
It doesn't denature the enzyme. Remember that non-competitive denatures it, because remember it goes in and it changes, kind of alters the shape of it. Okay? What are the things that affect enzymes?
Temperature.
Temperature,
pH.
pH,
pH.
and concentration. Remember? That's how we were able to deduce what the lab might be, because of we know that temperature, pH, and concentration are things that affect enzymes. All right? So we're good on enzymes.
Yes, ma'am.
All right. The next thing we're going to do is we're going to look at protein synthesis, and let's review a couple of things. Everybody got them off the board?
Yeah.
Yeah.
Oh, let me review something. How do you know if it is, if I gave you, remember we did this? And how it's drawn on your paper, where remember this? We know this is alpha because of the directions of how the oxygen is going down. Remember that if the oxygen was going up and connected this way,
That'd be beta.
that would be beta. Remember? Or sometimes it show it like a funky flip looking. Remember that would be beta just by looking at it. Alright? Now, let's review a couple of things. What's the difference between DNA and RNA? Give me some basic differences between DNA and RNA.
DNA has 2 strands?
Yeah, you are.
RNA has uracil.
And DNA will have adenine and thymine.
RNA is a single strand, while DNA is a double strand.
Isn't TH?
It's TH.
TH. Thymine.
What else? Ooh, where's this one found?
And the nucleus.
Can it leave the nucleus?
No.
No.
And RNA has like mRNA and tRNA.
mRNA. RNA can leave the nucleus.
Yeah, RNA can leave the nucleus.
And it can be found on ribosomes or in the cytoplasm.
Where else besides ribosome cytoplasm? One more structure.
Endoplasm.
Which one?
The soft. Rough.
Rough because it has the.
Remember it's found on ribosomes, messenger RNA, because it can be found on the rRNA. What occurs here with DNA? What process occurs with DNA? Well, yeah, mitosis. But remember, when DNA is being copied, what is that process called?
Synthesis?
Huh?
Synthesis?
No, is it?
When it's being copied, what is it specifically called? Transcription. That copy, when it goes through and it reads it, what is that copy making? What results from DNA being transcribed? What's the end result from DNA being transcribed? What's the product you get?
The identical strand.
Isn't it like a sister strand?
mRNA. Remember, when DNA is transcribed, at the end, the product is mRNA. And as Ellie said, that's a complementary
Strand.
It's a complementary strand or copy of the DNA. Because remember, one of the questions that you guys, and it's been on the test several times, they'll ask you, what's the product of transcription? And you guys missed, I put it on the test this year, remember? So as you guys are studying, you need to go back and look at your past tests. But they'll ask you, what's the product of transcription? And all it's asking you is, at the end of transcription, what do you have? You have that copy of messenger RNA, right? So the messenger RNA, what is its function? Why do we need messenger RNA?
To transfer signals or messages to.
Why did we make this copy? Because DNA can't leave the nucleus. Where are proteins made at? Where are proteins made? On ribosomes. If it's in the cytoplasm, it's made on ribosomes in the cytoplasm, right? So you have to remember that the reason, the whole purpose of this is so that that hereditary information can go to the ribosomes wherever they are, whether they're on the rough ER, or in the cytoplasm. It has to put together those proteins. Okay? So the whole function of this messenger RNA is so that we can get that information from out of the nucleus. Because remember, DNA is very fragile. It does not live outside of the nucleus long. It denatures very easily. Okay? So, we have messenger RNA. That messenger RNA makes a copy or complementary strand of the DNA. Where does it go from there? Where does the messenger RNA go once it leaves the nucleus? It goes to the ribosomes, right? So we have our DNA is being made into a complementary strand of messenger RNA. This process is in the nucleus. And that's transcription. Okay? Y'all know I love flowcharts, right? So the messenger RNA then goes to the ribosomes. What happens here?
Becomes tRNA.
All right? Is the T strand of tRNA exactly the same as the strand of messenger RNA?
No.
What's the difference?
Yourself.
Okay, well, we knew we got yourself here.
Yeah.
But what's the difference between this strand and this strand?
The basis.
So it's also a complementary strand of the messenger RNA. So we're going to do this just so you guys know. Okay, remember these three things, when we read them in threes, they're called codons. Right? And remember, codons make up parts of protein, and then it tells us, all right, so if this was my DNA, my complementary strand of this would be GTC, correct? Right? That's my messenger RNA. I know you guys remember this, we're just reviewing. So then the complementary strand of tRNA would be
C, U, G, CUG.
Is it C?
Isn't it A?
Yeah.
Why is it A?
Because T and U switch.
So then they become GUC, or is this?
No. Then if we made it into the rRNA,
It would be
It would be
GUC. Okay.
yeah. Okay. All right. And then remember, from this part, we're starting from here down when it makes this complementary strand, and then it goes on to the ribosome. On the ribosomes, the proteins are starting to be packaged. Right? remember, if they give you the universal code for, remember the little chart I gave you guys, where it tells you like CAG might be like serine or, you know, arginine or something like that, and then you build those proteins to make up whatever you need to make up. So let's look at the question. It says, name the stage of the mitotic cycle when replication of DNA occurs.
I thought it was telophase.
Transcription?
Huh?
Transcription.
No, we want mitosis.
I thought, wasn't it telophase?
Okay, first of all, remember we have
Oh.
I was gonna get it. It just took me a second. All right, remember this. Remember PMAT,
Yeah.
but you have I PMAT.
Mhm.
So this is interphase.
Prophase. Metaphase. Anaphase.
All right. Interphase is the longest phase of the whole cell cycle, right? What happens during interphase? Why is it the longest? What is the cell doing?
Copying the DNA.
It's just chilling.
But that's its whole life, and then
Then what triggers it to say, oh, I'm done chilling. I might need to work now.
like an outside signal.
So some signal that scientists have not yet to discover triggers it to do what?
Replicate.
Starts what? So what is it going to replicate?
The DNA.
Does it only replicate DNA? What's being replicated?
So-
Everything.
Everything. But during this phase, DNA right here. And interphase, that's the phase where at some point something says to it, oh, you might want to get a job. Okay? You might want to start doing stuff. Not only does it start to replicate the DNA, but it makes copies of everything. And I think that's what I think I didn't stress that enough when we originally talked about it. It makes copies of everything, not just the DNA. It makes additional copies of the endoplasmic reticulum. It's making new copies of the Golgi apparatus. It's making copies of everything. Because remember, in mitosis, we're taking one cell and we're making two daughter cells or two identical cells with it. So during interphase, this is when all of that stuff is being copied. So what happens during prophase? Everything gets copied. So now what happens?
So it's moved to the different poles.
No,
It-
that's not yet.
Not yet.
They find pairs?
Remember this?
Then
Yeah, remember? Yes. Chromosomes start to condense. Because remember, DNA is being copied and it has to be unwound in order for it to go along and read it. But because DNA is so big, we can't leave it like we have chromosomes, because if you literally took out all of your DNA, you probably can stretch it across this well, no, probably you can't. You can stretch it across this campus, the length of it. So now we need to take something that big and to condense it in something that we can't even see with our naked eye. So during interphase, that DNA unwinds. So during prophase, it has to condense back together. So during prophase, and then yes, the centromere is starting to be made and things like that. The spindle fibers start to form. But the big thing is you need to know that those chromosomes start to condense. Then what happens during metaphase?
They line up. They
Yeah,
met.
remember meta as middle. Remember? They line up in the middle. Why are they doing that?
So they can split apart. They can split apart.
so they attach to the spindle.
So they attach the spindle fiber. What else is the cell doing? As it is attaching them to the spindle fibers in order for them to...
Or like
Huh?
disappearing. Everything is make sure it has enough them.
It's checking to make... This is during the time of the cell cycle where the cell is going through to make sure it has a copy of every chromosome that it's supposed to have. That's why they line up in the middle. They line up in the middle so they can attach to the spindle fibers. But also, so the cell goes through a check. Do I have 2 chromosome number 1s? Do I have 2 chromosomes? So it makes sure that it does. During this time, normally the cell will catch anything that's been copied wrong or any mutations.
What happens if it does?
Huh?
What happens if it does?
Then the cell doesn't. Then the cell typically it should die. So if it's going through and it says, "Oh, I have an extra set of chromosome 21," it should, in theory, die because it's an abnormal cell. However, we have cancer and we have kids walking around with Down syndrome and other hereditary diseases because they have extra chromosomes or they don't have enough. So in some way, during this process, something didn't tell it, "Oh, this is not right. This cell needs to die and we need to get rid of it." Okay? So it's doing a spot check to see, "Oh, I have everything. Oh, it's abnormal. Get rid of it." Okay? So then what happens during anaphase?
The spindle fibers just split apart.
The spindle fiber starts moving the chromosomes to opposite ends of the pole. Why?
To prepare it to split into.
Is the chromosomes the only thing that's being moved to opposite ends? What else is being moved?
It's like pinching points.
The cytoplasm.
What?
Organelles.
Why are the organelles being moved?
So that when it splits, each cell has enough.
So during this process, because remember I said during this process, not only is it replicating the DNA, but it's making everything else. So also during this process, not only are the chromosomes moving, but it's also making sure, "Oh, do I have enough mitochondria here? I need some mitochondria there. I need some Golgi apparatus." So it's making sure everything is dividing. That's the one thing that Cambridge does when it goes from ICSE to AS. You need to know that. Not only is the DNA being moved, but everything else in the cell is being moved, okay, or being divided, all right? And then, what happens during telophase?
The spindle daughter cells.
It starts to pinch, right? The cytoplasm starts to pinch. Let me explain the reason why I broke it up to you guys for this way. This is mitosis. During mitosis, the whole goal of mitosis is for the nucleus itself to divide. During cytokinesis, think of cyto as cytoplasm. During cytokinesis, the cytoplasm is starting to divide, and that's when the new cells are formed. During telophase, here, it pinches off, but the important part here is that the nuclear envelope starts to reform again. Because remember, here it had to start dissolving, right? Because it has to be able to, DNA has to be able to get out. But at some point during this, because the whole purpose of mitosis is for the nucleus to divide, at some point during this time, the nucleus has to reform. And that's what happens during telophase. A lot of people think telophase is the end and the cell has divided. It hasn't. Just the nucleus has finished dividing. During cytokinesis is where the cytoplasm divides and the cell membrane or cell wall reforms. And you have your two new cells. Okay, good overview, right? I don't have to draw the pictures because you guys have that. All right, so the next question is, name the stage during the mitotic cycle when DNA replication occurs. That will be during interphase. And we had to go through all of that just to get the answer of interphase. But I wanted to review. All right, name the bonds shown by figure 5.1, the dashes. If you look at it, it's connecting the uracil, the uracil, the thiamine, I just forgot what they call base pairs. It's connecting the bases. So the bonds that connect the bases together are what type of bonds?
Hydrogen.
Hydrogen bonds. Okay. And it says name the bases of M and O. How do we know that? How do we know what it is?
By the shape,
So you need to know what they look like, right?
shape.
So what's M?
Thymine, no.
Adenine.
Adenine, but you also could have knew because it's attached to that thymine, right?
Yeah.
So what's O?
Cytosine.
Cytosine. Because remember,
M is adenine.
this is in DNA, in RNA,
And O is that's O.
huh?
Cytosine.
It says, explain why DNA replication is described as a semi-conservative process. Why is it semi-conservative? Misa?
Half of the original molecule is kept.
Yep, because you are making DNA acts as a template. So you're keeping, you already have that template, so half of it is originally same. So it has some of the old. So the reason why it's considered semi-conservative, because you still have DNA, you're using that strand of DNA as the template, and once it's copied, you still have parts of the old. Even if by some chance some mutation is inserted, you still have some of the original. All right? And then the enzyme that catalyzes the replication of DNA checks for errors in the process and corrects them. This makes sure that the cell produced in mitosis are genetically identical. Explain why checking for errors and correcting them is necessary, which I already just told y'all, but tell me why.
No mutations.
No mutations. What else?
Mutations result in cancer.
So no cancer. What else?
No Down syndrome.
Okay. So this is what you could have put. It checks for mutations. It makes sure that the production of proteins isn't altered. So it makes sure that what you started with is what you're going to get in the end. So it's going through to make sure that something wasn't copied wrong. Where there should have been an adenine, then it was copied wrong. It has to be copied right, because remember, if it's copied wrong, what happens? What is your body supposed to do to the cell?
Kill it.
Kill it. So that you don't have,
Mutations.
yeah, so that the mutated cells are destroyed, so that it prevents cancers or anything like sickle cell anemia or any type of conditions, monogenetic condition like that. And it helps to make sure that every cell is programmed to die. Because when it goes through the check, every cell has an expiration date. If a cell doesn't have an expiration date, and if it doesn't go through AICE, which is the death, the process of a cell dying, then AICE, I don't, it's APOPTOSIS, AICE, but it's the death of a cell. So it makes sure that it dies. Because remember, means without. All right.
Is it?
Good?
Yeah.
Yes, Jonathan.
I just wanted to point out that
No.
number 5 BI, the one where it's asking about the bonds between, those are hydrogen bonds, but the bonds that actually make up the backbone itself is called the phosphodiester bond, because I know that they've asked that before.
Yeah.
There was some confusion about which type of bond that was.
Yeah, the bond that makes the backbone?
Yeah.
Yeah. It's phosphodiester bond.
So, would either of those work as the answer?
No. You need to know that the bonds between the bases are hydrogen. The bonds that hold the bases together are hydrogen bonds.
But on the back.
The bond, yes,
Okay.
it's phosphodiester, or it's P.
Here we go. Not even sure.
Right? No, I spelled it wrong?
PHS.
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 No, PHOS. Yeah.
Oh.
PH.
pH again?
Yeah. Phospho.
Oh, yeah.
And then diester.
It's a big difference.
Boy, this like
Oh, phosphorus.
Alright, anything else when it comes to proteins? I don't need to go through the whole, give you a sample one and you make it. You guys remember that, right?
Yeah, that's easy.
Yes, Misa?
Is this the one with the shapes, like the amylose? When you do
Pentagon of death.
this one, like the top one,
When they were named.
I'm sorry, I can't see that far.
like the amylose and cellulose with the shapes. Those are these. I have like I don't remember what they look like.
Oh, that's, you're gonna have to make flashcards, because there's no trick that I can, you know what I'm saying?
Pentagon of death.
Yeah. Remember, there are certain ones you just need to know. Like, you need to know what glucose looks like. The ones that we went over in your IMB that I had you look up, you need to know those, I think it was three or four?
Three.
Three of them? You need to know what those look like.
If they throw one at you that's only like, yeah, then that's gonna be a reducing sugar.
Alright? Anything else about cell division? Because we kind of, we didn't go into that in detail in here because we went over it freshman year, and you go over it every year in science? Every year?
Great.
Well, that was different. You guys had
Chemistry.
chemistry, physics. You shouldn't go over.
What is that?
Alright, so we're good.